This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode number 365, recorded on November 27th, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, Vincent.
Happy Thanksgiving.
Happy Thanksgiving to you, too.
Yes, the post-Thanksgiving Friday here in the U.S. It's a holiday we celebrate, but not everyone around the world does, of course.
It's also Bill Nye's birthday today.
How
Bill
about
Nye,
that?
the science guy, yeah.
Neat. Well, happy birthday, Bill Nye.
Yeah.
And it's you and me for now. Alan is on the road. Maybe he'll connect, or maybe not, but we can talk about viruses, right?
Sure.
This episode of TWiV is brought to you by ASM Microbe 2016. This is a combined meeting of the previous meeting known as ICAC and the main general meeting of the American Society for Microbiology. So in 2016, they're doing what's called co-localization. It's going to be in Boston, and you should submit your abstract now. Boston, Massachusetts, the best microbial sciences in the world, and you can go there and explore the full scope of microbiology. This meeting provides a distinct forum for cross-disciplinary study, both in the classroom and in the integrated exhibit and poster hall. The meeting is designed to spark collaboration and discussion. So remember, ICAC, Intersciences Conference on Antimicrobial Agents and Chemotherapy. There you go. It was sort of the clinical meeting, and the ASMGM was mainly basic microbiology, but a little clinical stuff thrown in. They're going to all be together at one huge meeting in Boston, MA. If your work touches on the microbial sciences, this is the meeting you should attend. Learn, connect, and advance at the inaugural ASM Microbe 2016. If you want to register and learn more, go to asm.org/microbe2016. And I will be there. I believe Alan Dove will come over. He's only about 2 hours away. And we are planning to record both a TWiV and a TWIM from the meeting. So check it out.
And then a few days later, you'll come to Virginia Tech.
That's right. Exactly when, I don't know yet, depending on... I asked them to schedule the TWiVs and TWIMs by Saturday. I'm going to probably miss the keynote. Mm-hmm. I have a suspicion that I will get there sometime on Sunday. Mm-hmm. It's not easy to get to, Blacksburg. For me, anyway. Blacksburg. Yeah. I have to fly to... Usually, I go to Dulles, and then I connect to... what's the city you fly into there? Roanoke. Mm-hmm. And then it's an hour drive, right? They're going to have shuttles running?
Yeah, they're going to have shuttles and buses, and I think it's about 45 minutes. Not quite an hour. Mm. And there used to be direct flights from some additional cities to Roanoke when they first put in the bid for the meeting to be there. But, for instance, there was one from Detroit to there that Thea had taken. And, oh well.
Yeah, I mean, would love it if there were a direct Newark, but that's not happening. There was a direct Newark to London, Ontario, a year before the meeting we had last year, and then they canceled it. Mm-hmm.
So I read an article that the airlines are canceling those kinds of direct things to small cities, so...
Wow, Alan Dove just signed on to the Google Doc, so he must be close by, right?
Mm-hmm. He texted and said 5 minutes, about 5 minutes ago.
Nice. Yeah, his face just popped up there, and we will add him to this conversation.
So, meanwhile, I can tell you about the weather here.
Oh, that's right. Tell me about the weather.
It's 45 degrees. It feels like 41. Humidity 94%. 8 miles per hour from the north, and it's that means it's 8 degrees Celsius. And it's been raining. I can't tell that it's raining right now, though. My windows are too dirty, and the puddles aren't very big. They're off in the distance. So But we did have snow last week, the day I came back from Washington, D.C., and then it warmed up and it's all gone now because it was a lot of snow. It was nice to have it go away.
Yeah, I heard that. I follow Rebecca Skloot on Instagram, and she lives in Chicago, and she had snow there.
Hello?
Hello, Alan.
Oh, there you are. Okay.
You must have muted me because I heard you before.
Yeah, I had just plugged in my headset, which of course I had packed down to my in-laws, and then it didn't recognize it, and yada, yada, yada.
How long does it take you to drive back?
2 hours.
I'm sorry you came back. Didn't you want to stay longer?
Oh, well.
Not really.
No.
Yeah, I don't blame you. Was everyone mad that you drove back in the morning?
No, actually, that was my wife's idea.
Excellent. That was good.
So she said, let's go. Okay, if we do it in the next few minutes.
Well, thank you. Good. You sound good.
Okay.
Yeah, probably would not have been as good from
No,
there.
they're technologically still stuck in the mid-20th century.
Yeah. Well, I'm home here, and I have you both on one channel because I don't have a multi-channel setup here, and so keep the extraneous noises to a minimum, please.
I'll try and mute my microphone before I burp.
And here the weather's beautiful.
It's gorgeous here, too.
Yesterday I went out without a jacket on. Must have been in the 50s. And it's clear skies and just beautiful today as well. So that cold weather is gone.
For now.
It should come back, right?
Probably.
But it's unusual for Thanksgiving for it to be this warm, right? All right, well, Alan, we didn't get very far. We just got to the weather.
Oh, okay. Well,
So
it's 66 Fahrenheit and sunny skies and calm winds here. It's gorgeous.
Nice. All right, we have a follow-up from, Ayinka. You think that's how you would say that?
Ajinkya?
I don't know if you pronounced the J or not.
I don't know either.
Ajinkya or Ajinkya. Either way, Ajinkya writes, Dear Vincent, I hope this email finds you well. I have been an avid listener of TWiV and enjoy your podcast very much. I am also a recent graduate of Northwestern University Chicago, graduated with a master's degree in biotechnology, currently working as a research tech at the Gottwein Lab at Northwestern University's Feinberg School of Medicine. I study KSHV and EBV herpes viruses. Although I was excited to hear that about your contest to give away free copies of Principles of Virology, I was also sad about the fact that you wanted more Twitter followers in return. Well, I am not on Twitter, trying to curb my social media addiction, and so will not be able to follow you, but I can tell you that the reason I listen to TWiV is to know more about the seemingly infinite types of viruses that I do not get time to read about, but more important than that, I listen so as to learn how to critically analyze and interpret data mentioned in a paper like you and Rich and Kathy and Dixon do. That's more than 140 characters, Ajinkya. Well, I figured Twitter was a good place to reach lots of people, but apparently we don't have so many fans there. You know, I had so many followers that I figured some of them were listeners.
And some of the people didn't fit their tweets into 140 characters either. There was one that was 5 tweets.
Yeah.
There was one that was 2 tweets. So, yeah, we can't pick on Ajinkya for that.
I mean, as I said, there will be other opportunities as well. And
I sympathize with this. I often see stuff that's, enter this contest by liking us on Facebook. Well, I'm not on Facebook.
Yeah, well, I have multiple copies, so we'll have multiple contests. I appreciate the time and effort all the TWiV hosts put into making episodes, and I feel like I must tell you not to be disheartened about the lukewarm response you received for the contest. I am certain about the fact that there are many like me who are not on Twitter, but appreciate your work nonetheless. That being said, I'd be delighted to get a copy of Principles of Virology and assure you that it would be put to good use. No pressure. Thank you for your efforts and keep up the good work. P.S. It's a cool 8 degrees Celsius here in Chicago with yet another Chicagoan winter on the horizon. All right. Well, it was, as I said, it was just the first try, and I know not every listener's on Twitter, but we'll move to other venues and other. In fact, today we'll have a different contest, okay? And we'll talk about it at the end. And you cannot fast forward to it. You have to listen to everything. You can do what you want, obviously, but
I guess we could scatter hints through the show.
Yeah, that would be fun. You could take you take all the sentences for the contest and one here, one there, and that way they couldn't go to a certain place. That would be creative.
Yeah.
All right, we have a couple of snippets. This first one was sent in by Steen, and this has to do with CPF1 priority patents, credits, and press releases, and it revolves around a very nice story in Wired by Sarah Zhang called "The Battle Over Genome Editing Gets the Science All Wrong." And the illustration, if you don't want to read it, just look at the illustration because it's cute. I like that.
Yes.
This has to do with the CRISPR system and the Nobel Prize and the patent and so forth. And so having read this, we're going to just chat a little bit about this whole system because it's pretty interesting. Now, we've talked about CRISPR-Cas9 before on TWiV, but just to sum it up very briefly, it is a system found in bacteria and archaea, which is an adaptive immune response, basically, which is a sequence-based immune response. When these bacteria receive, say, a phage or foreign DNA, they chop it up and put pieces of the sequence into their genomes, and then the next time they are infected, they produce small RNAs from these templates in the genome, and those together with nucleases chop up the incoming DNA. And so many bacteria and archaea have many arrays of these sequences representing different insults they've had before. It's very cool. And it's become really hot now because you can utilize the sequence-based clipping to edit genomes. And, we've had other ways to do this, like TALEN nucleases and small interfering RNAs. But this one shows a lot of promise. It's relatively easy to do. We're doing it in my laboratory, for example. And
so it must be easy to do.
I was going to say that, actually. Yeah. So it's relatively simple. Now, the problem is that, of course, it's so hot because of the genome editing. And remember, we've talked on TWiV about editing humans and so forth. There's a lot of controversy, but there's also a lot of promise and a lot of money involved in this.
Huge
sums of money. And so the main players that you have probably heard about are Jennifer Doudna, who's at UC Berkeley, and Emmanuelle Charpentier, who's now at the Max Planck Institute for Infection Biology. They have published a main paper, which we'll talk about in a moment. And then you have a molecular biologist at the Broad Institute up in Boston, Feng Zhang, who owns the patents for CRISPR-Cas9 and says he came up with the idea independently. And the gist of this article is that if you're trying to decide who is going to get the Nobel Prize, they're arguing over the patents, of course, already, because Doudna and Charpentier said they did it first and they should have had the patent. But in fact, this thing was discovered many years ago, and I like what they say here. Casting the narrative as Doudna versus Zhang or Berkeley versus MIT is a misapprehension of history, creativity, and innovation. Discovery comes not from a singular stroke of genius, but an incremental body of research. And in fact, if you look at the history of CRISPR, it was first described in 1987, the CRISPR array, an array of sequences in E. coli. 1987. So these are people working in prokaryotic systems. They find something interesting and they pursue this. And this is known as serendipity in science. We talk about it all the time. And it's one reason why it's hard to do risk-benefit, or at least benefit analyses, because you never know where things are heading. So, in 1987, they publish a paper. And let's see what the name of that paper was. Nucleotide sequence of the IAP gene responsible for alkaline phosphatase isozyme conversion in E. coli and identification of the gene product. And buried in there was the fact that there were arrays of these short nucleotides, right? And from there, in 1987, people started pursuing it. They found these sequences in many different bacteria and archaea. In 2002, they found a series of genes associated with CRISPRs. So if a genome didn't have a CRISPR, it didn't have these other genes. They were called Cas for CRISPR-associated proteins. And CRISPR, of course, is clustered, regularly interspersed short palindromic repeats. CRISPR-Cas. Cas is a complex of proteins that does the processing of the target gene that's incoming with the little RNAs that are produced from the array. So 2002, the Cas genes were identified. Okay, now...
And that, by the way, these papers, this 1987 discovery, this 2002 paper, these are all people other than the ones who
exactly
just listed as being associated with this.
Exactly. This is like restriction enzymes. Some people discovered the phenomenon of restriction, which means when a foreign DNA comes into a
little of this,
bacteria, it chops it up and eventually becomes the biotechnology industry or a main part of it. Right. And other people get in at the end. So now in 2007, a science paper, again, by none of these individuals, first author is Barong Zhu and the last is Horvath, CRISPR provides acquired resistance against viruses in prokaryotes.
And by the way, that group, I think this whole story is actually also a really good illustration of why you can't say, oh, well, applied science does all the good stuff, so we should focus on translational research and applied science because basic research is just intellectual masturbation. But if you look at the history of this starts out as a really, really basic research question.
Yeah.
How do bacteria resist phage? Well, who the heck cares, right?
Mm-hmm.
That's not applied research. It's totally basic, curiosity-driven stuff. And really one of the most basic papers in it is this one, this Barangu paper. And by the way, these researchers work in industry.
Mm.
They work for Danisco, which is a yogurt company.
Yes.
And they're studying resistance against viruses in prokaryotes because they're interested in prokaryotes because they work for a yogurt company.
Yeah.
So even the corporate world understands the importance of basic research, and yet we're going to get to an endpoint that is extremely application-oriented.
Yep. So now a name comes into this, which I had not heard of. It's part of the story from the Wired article. Virginia Siksnis, a molecular biologist in Lithuania at Vilnius University. He got interested in CRISPR in 2007, and he says here, in my lab, we didn't know how to make cheese or yogurt, but we know how to work with E. coli. Mm-hmm. I think that's great. Mm-hmm. So he started working with this, and he found which associated genes were important, and found one, Cas9, which is one that's often used today in the applications, that was important for snipping the DNA of the incoming DNA. So they submitted a paper in May 2012 to PNAS, okay? Took a couple of months to get published, but a month after his group submitted that paper, Doudna and Charpentier's paper came out in Science, all right? And that one was fast-tracked because Science recognized that this was a big deal. It's called a programmable dual RNA-guided endonuclease in adaptive bacterial immunity. Now, these are functional advances. They're showing exactly how the system works. Obviously, they're important. But in the gist of the Wired article is that Siksnis is probably going to get left out of all of this just because his paper took longer to review.
Yeah.
Right.
But he made a key finding as well. He found Cas9 in E. coli. And as far as I can tell, he doesn't seem like he's in the running for a Nobel Prize. He certainly doesn't have the patent. And as they say in the article here, only 3 people can get a Nobel Prize. So I would guess Doudna and Charpentier are likely to get it. The fellow at the Broad, Zhang, they published papers in 2013 showing that you could get CRISPR-Cas9 to work in human cultured cells. So that's important also, right? That's what many people are using it for. And he got the patent for that.
Which is interesting because given the 2012 Charpentier, Doudna work and Siksnis work that all came out before that, you'd think somebody could make the obviousness argument. Right? A patent has to be non-obvious. Well, we've got this editing system now fully described. What's an obvious thing to do with it? We'll demonstrate that it works in human cells. And I guess Zhang's argument on that would be, well, nobody had actually demonstrated that. So to do that was the patentable invention. But, patents are not real until they're challenged in a court and proven. So it is still possible that somebody could challenge that patent. It may not be enforceable. I don't know.
Yep. Anyway, he got it, and Doudna and Charpentier, or maybe Berkeley, are challenging that, I suppose. And Zhang, of course, has got the patent. So I don't know who's going to get the Nobel Prize. It seems to me that sickness should be part of that. He made an important discovery. Mm-hmm. Personally, I would give it to Doudna, Charpentier, and sickness, right? And I would say Zhang did what you said, Alan, is a pretty obvious experiment, right? Everyone else figured out what this does in bacteria. You just put it in mammalian cells.
Right.
However, Marty Chalfie got the Nobel Prize for putting GFP into an animal, right? And others had discovered GFP. So, there's Nobel precedent for that. I don't know.
Well, the Nobel Prize itself, I mean...
Yeah, I have my notes here. These are all from last week. We should get rid of the Nobel Prizes and give the money to R01s.
Right. Except, I mean, this is basically the Nobel Prize was a program to assuage the conscience of an arms dealer. This is somebody who invented a way to blow people up, and after profiting immensely from that, was racked with guilt and decided, well, I really ought to try and celebrate something that's good about humanity instead of just all this bad stuff. And so he came up with the Nobel Prizes, and that has had a distorting influence on science ever since.
Yep.
So we're going to have a listener pick about this, but from watching that pick, I figured out that there is this International Summit on Human Gene Editing that's going to be at the National Academies December 1st through 3rd. It's too late for the public to register, but it's not too late for the media to register, Alan, if you wanted to go. But anyway, the chair of it is David Baltimore. And the first speaker is Congressman Bill Foster from Illinois, and he's a physicist by training. A good friend of mine was in school with him for a while. So, and then there's various other people listed on the agenda.
Nice. By the way, go ahead. Sorry.
I was just going to say, so they're going to talk about, the ethics of this. And it's a global discussion. They have people from the Chinese Academy of Sciences, the Royal Society from the UK, and from the United States. So go ahead, Vincent.
So the way we are using this in my lab, and which is a typical application, you have a cell line where, say, you want to disrupt the gene. So you introduce the Cas9, a plasmid that produces Cas9 into that cell line. So we've done this for HeLa cells. Now they produce Cas9. And then you simply introduce 2 oligonucleotides that target your DNA sequence, and you can put them in on a plasmid as well. And then they go in the cell, and the Cas9 complex takes that and finds the target and chops it up. It makes a break, and that gets repaired, and your gene is disrupted. So it's pretty cool. Mm-hmm. And it's straightforward to do, right? It's very simple. Now, Steen also sent us a couple of other links. He sent us a link to a nice review article about... So the CRISPR-Cas system is much more complex than we have led you to believe here. And in fact, another RNA-guided endonuclease besides Cas9 now has been found by the Feng Zhang's group, CPF1. And as someone mentioned, now this is entirely his, so if he wants to patent this, he could do that as well. He's not sharing that with anyone because he discovered CPF1. And the other article, and I'm sure there are many more to be discovered as well. It's probably more complex than we think. And the review article also gives a nice overview of all of the different systems. It is by our friend, one of the authors is Eugene Koonin, the last author, an updated evolutionary classification of CRISPR-Cas systems. And, it's very interesting that all the relationships between the Cas proteins and he tries to put them all together into an evolutionary model in bacteria and archaea. They're probably both behind paywalls, unfortunately.
Yes.
But we will put links nonetheless, and someday we will have a detailed paper discussion of CRISPR, I'm sure. But thank you, Steen. That's cool. And I learned about sickness, and I hadn't known that before. I'd known about the Charpentier-Doudna-Zhang battles, but... he should be part of it. But from the quotes they have of him, he seems maybe he just likes his science because he said, that's the way that it falls,
Yeah.
It's good.
Right, but that's the thing with these discoveries and the structure. Well, the Nobel Prize really just echoes this common fallacy that we have that the great man theory of science and history, and there's one person or there are two or three people at most to credit for these great breakthroughs, and that's hardly ever the case. These are things that were due to be discovered, and the technology reached that point, and the knowledge reached that point, and a bunch of people found bits and pieces of it, and then it all came together, and a few lucky ones happened to be in the right place at the right time. I mean, not to take away from what Charpentier and Doudna and Zhang did. They did a lot of hard work, and the reason they were in the right place at the right time is because they were thinking the right way. But to single out a couple of people from that group and say these are the discoverers is just really misleading.
A couple of months ago, maybe 6 months ago, Jennifer Doudna won a prize for this. And someone tweeted, Jennifer Doudna wins this prize for discovering CRISPR-Cas. And I tweeted, I said, no, she didn't discover it. She figured out how it works in many ways. It's important. But discovery goes way, way back. And just let's remember that a lot of things have to go on. I mean, if you just look at the discovery of reverse transcriptase, Nobel Prize for Baltimore and Temin, right? They built on a lot of work that was done for many years beforehand, but some people make the Eureka discovery, and that's fine. But, I just don't think the Nobel is a good motivator. I think it really warps the way science is done, and it's just best for people to do experiments.
They stood on the shoulders of giants.
They did.
To use the words of somebody who was not normally known for his humility.
Yeah. Is that Pasteur?
No, that's Isaac Newton.
He said that. Oh, yes. Well, he was not known for humility. I didn't know him.
Oh, well, apparently he was a real arrogant something or other.
All right.
But even he was willing to admit that his work was not in a vacuum.
We have one more snippet. This is from Anthony, who simply wrote, "Dear TWiV panelists, where did this virus originate from?" And Tony sent a link to an article published in MBio. And Kathy's going to tell us all about that.
Sure. So the title is Virome Analysis of Transfusion Recipients Reveals a Novel Human Virus that Shares Genomic Features with Hepativiruses and Pegiviruses. And the authors are Kapoor, Kumar, Simmons, Buva, Chauhan, Lee, Saul, Jin, Morse, Shaz, Burbalow, and Lipkin. And you may remember that we've had Ian Lipkin on TWiV, I think maybe 4 times before. And Amit Kapoor has actually been on twice before also, TWiV 231 and 137. But since they submitted this paper, September 2nd, he's moved to Ohio State. And so I wrote to him to get a little background. He had done his PhD on polio and non-polio enteroviruses in India, and then he did a postdoc with Eric Delward at UCSF, where he identified a big group of new picornaviruses and astroviruses. And then he moved to Columbia in 2010 as an assistant professor, where he identified several viruses, including the animal homologs of hepatitis C virus. So his new position is principal investigator, and he's at the Center for Vaccines and Immunity of the Research Institute at Nationwide Children's Hospital, also the Department of Pediatrics and the medical school at Ohio State. So what they did in this paper was look to see whether there's novel or as yet unknown viruses that they can identify in blood transfusions. People who've gotten blood transfusions. And so they take advantage of this really cool biological resource, an NIH sample repository at the NHLBI, one of the institutes at the NIH. And in that institute, they have a sample collection of transfusion-transmitted viruses. Well, the name of it is Transfusion-Transmitted Viruses Study, and they have samples collected pre- and post-transfusion from a variety of people from 1974 to 1980. And so what's the thought process behind this? Well, back before we had screens for some viruses like hepatitis C, hepatitis B, and HIV, these viruses ended up in hemophilia patients who get lots of transfusions or other people that get transfusions. So it's really important to screen blood and blood products. And if you don't know what viruses are there, you may not be screening for all the viruses that you might want to screen for.
And that's a really prescient set of samples. I mean, when you think about they were collecting those and storing those, and somebody thought to do that before we knew about HIV, you know, that just really I think that deserves to be called out as someone ought to get some credit for having put those aside.
Definitely.
It's a very smart idea.
And you know, for the Eric Delworth's lab is in the blood center over there in San Francisco, and so Amit, I'm sure, learned about or thought about blood a lot while he was working there, because they do screen. They have this wonderful resource, and Rich and I visited years ago. In the basement, they have freezer after freezer after freezer with many, many, many samples of blood that they've stored over the years, and it's a resource, incredible resource that you can go back and do these kinds of studies with too.
Yeah, so just to make a little point of this, the sample collection lasted until June of 1980, and the first AIDS case in the United States recognized at the time was in April of 1980. So this is pretty much before it was known that there would be HIV in blood products. Now, in retrospect, people have gone back and figured out that some deaths were due to HIV because they had tissue samples and they could assay for that now, but as Alan said, it was really a brilliant stroke to do that. So in this study, they took advantage of these matched samples, and they did deep sequencing. And the bottom line is that they identified a new virus that had characteristics of viruses from 2 genera in the flavivirus family. So I'll be rich here for a minute. Flaviviruses, plus-strand, single-strand RNA enveloped viruses. And so the 2 genera here are the Hepativiruses, where there's hepatitis C, and Pegiviruses. And examples of Pegiviruses mostly have these GB names, like GBC and so forth. Anyway, so for a long time, the Hepativiruses were thought to be mostly or exclusively human. And then Amit and Ian Lipkin and others identified other Hepativiruses in other species. And then this other genus, the Pegiviruses, it's known that there's human Pegiviruses, and it's found in about 1 to 4% of healthy blood donors, and it seems to persist for years with as yet no apparent symptoms. Okay, so they got 44 samples or subjects where they had the pre and the post-transfusion samples, and they did deep sequencing. The method that they used would actually allow them to get DNA viruses and RNA virus sequences in their deep sequencing method. And the first thing they noticed is that they had lots more sequence reads, both in total virus sequences and a particular Anello virus group of viruses. They had more of these sequences in the post-transfusion samples. And just now, as an aside, I'll be rich again and remind you about Anello viruses. We had XJ Meng tell us about those on TWiV 285. Anellos have circular negative strength DNA genomes. They're pretty short, around 3,000 to 4,000 nucleotides. It's a pretty recently discovered family, non-enveloped, and they cause chronic human viral infections, again, also not particularly associated with any disease that we know of. And these are the ones that Vincent really likes to say the name of. So, Vincent?
Torquitano virus.
Right.
Love the name.
And Torquitano midi-virus and Torquitano mini-virus. So, anyway, so they found a lot more of those sequences in the post-transfusion samples. And so then they decided to focus on 12 subject pairs in total, 6 where the Anello virus reads didn't vary very much between the pre- and post-transfusion, and 6 subjects where they were 10 times higher in the post-transfusion set. And they did a really detailed comparative analysis. And they show this in Figure 1 in an interesting presentation where they list all of these viruses vertically, and then they have these dots going across for the various 12 samples, and the size of the dot reflects the size of, or the number of sequences that they got. So the bigger the dot, the more sequences they got. And so the right half of the picture shows the individuals sample, the 6 individuals that they chose that had 10 times more sequences after transfusion. And so that's pretty easy to see just from a distance. And they found that there were new Anello viruses, but they also found that there were a lot of Anello virus sequences that were the same between pre and post transfusion. Okay, so one other thing, just before I get to the main part, at the very bottom of this list, there's one human immunodeficiency virus sample that they found post-transfusion.
Yes.
So I asked Amit about this, and he said, well, we didn't confirm it by PCR, and so not everything you find in high-throughput sequencing is real. But that would have been a sample prior to one of those samples that now in hindsight we would say, was potentially HIV positive. But that wasn't the aim of their study. He also mentioned that they found a couple other DNA viruses and things that aren't the focus of this study. So they had 40% of the sequences that remained unclassified, and they had low similarity to any known viruses. And so they analyzed these further, and they found this novel flavivirus in 2 samples, 2 post-transfusion samples. And this virus had distant homology to both hepatitis C virus in the Hepaci genus and the human Peggy virus in the Peggy virus genus. And so they called this virus human Hepeggy virus. It's the hep from the hepatitis and the Peggy from the Peggy. So from here on, we're going to call it human Hepeggy virus. And then they got the complete sequence of it, with which they could then do a lot of things. And so, as expected for a flavivirus, they found one long open reading frame. They easily identified the termination codon, but there was a bit of a quandary because there's 5 potential AUG initiation codons for translation. And they've narrowed in on the third one for 2 reasons. One is that it's really the only one that's in an optimal sequence context for translation. It's in the best Kozak-like sequence. And also, when they determined the sequence of the 5' untranslated region and its secondary predicted folding structure, the place where this third AUG is optimal relative to the internal ribosome entry site, the iris site. So they haven't proven it by any means, but that's their reasoning by which they say they think that third AUG is the one that's used. They did sequence alignments of the human Hepeggy virus with hepatitis viruses, hepatitis C viruses, and Peggy viruses. And they show in the paper comparison between 2 highly conserved genes, NS3, which is a component of the protease and helicase, and NS5B, which is the RNA-dependent RNA polymerase encoding region. So these regions would be highly conserved, as you might imagine, because they're performing important enzymatic functions for the viruses. So they compared those, and they got similar phylogenetic trees with both regions, the NS3 and the NS5B. And they found that the tree has a Hepacivirus clade, or branch, if you will, and a Peggyvirus clade, or branch. And the Peggyvirus clade has 2 main sub-branches called 1 and 2. And for both of those genes, the NS3 and the NS5B, the human Hepeggyvirus falls on its own branch in the Peggyvirus group 2. But it's not very close to any of the other ones in the group 2. Those are rodent and bat sequences. So we'll come back to this. We're going to kind of keep a tally of what features this virus has that are Hepaci-like and what features it has that are Peggy-like. So far it's maybe a little more Peggy-like. And then they used an algorithm to predict...
Which is good if you're getting worried about this virus.
Right. So they used an algorithm to predict the protease cleavage sites of this polyprotein, and that led them to predict that there's a short 65 amino acid cysteine-rich protein at the very 5' end of the encoded in the very 5' end of the RNA, and they called it protein Y. And some similar proteins have been seen in bat and rodent PEGI viruses. And among them, they don't have any sequence similarity per se, but they're all very highly basic. And this protein in the human HPEGI virus is also basic. And so that makes it a little bit PEGI-like. Right, okay. And then they looked at the HPEGI virus E2 envelope protein and found that it's predicted to be highly glycosylated. And that makes it more like Hepatitis viruses. And then, as I said, they looked at the 5' untranslated region, and they looked at its predicted secondary structure, and it matched the other group 2 PEGI viruses, making it seem kind of PEGI-like, but it also had motifs with similarity to the Hepatitis virus 5' untranslated regions. So that makes it a little mixed bag. Maybe it's a little more like a Hepatitis again. And they did note that unlike the Hepatitis, it doesn't have a seed match sequence for the miR-122 microRNA for the cognoscenti that might be interested in that. Then they used some program where they could predict the thermodynamic folding of the whole genome, and it's in all caps, GORS. And what they found was that the human HPEGI virus has a large-scale structured genome that's pretty much like the group 2 PEGI viruses. So scorecard so far, genome sequence, more PEGI-like, that short polypeptide, PEGI-like, glycosylation of E2, Hepatitis-like, 5' UTR, Hepatitis-like, the GORS, PEGI-like. And as we're going to see in just a minute, it has persistence in the population in these samples, but that doesn't make it either particularly PEGI-like or Hepatitis-like. So having had these samples from these 2 individuals out of the group that they looked at, they went back and looked at more samples from the transfusion study, 46 pre and 116 post-transfusion studies. And the first 2 that they got were all that they got. And so it's not all that prevalent. We'll come back to that in a minute, too. So 2 instances out of 116 post-transfusion samples. When they found it in these post-transfusion samples, it was either 7 or 17 days after the transfusion, and then 240 or 280 days later, it wasn't in that same individual anymore. So evidently, it causes a transient viremia, but then it gets cleared. So then they used another resource that they could get from the NHLBI, which is a multi-center hemophilia cohort study. And those samples were collected from 1982 to 1986. And as I mentioned before, hemophilia patients get a lot of blood product transfusions. So this is another likely source. They found out of 106 individuals that they looked at, their samples, they found 2 positives there. So again, the frequency is something like 2 out of 106. One of the individuals, all 4 of the longitudinally collected samples were positive, spanning more than 5 years, so persistent over a pretty long time. And then the other individual had 4 samples. The first one was negative, the last one was negative, and the middle 2 were positive over about a 200-day span or more. So again, it looks like these viruses can persist for some length of time, but they may be able to be cleared. They also noted that of these 4 subjects now that were human HPEGI virus positive, 3 of them were positive for hepatitis C, and all of them were negative for hepatitis, for the PEGI virus, excuse me, the GBV-C. So the fact that so many of them were positive for hepatitis C, again, remember that this is samples up until the 80s, and I don't think there was probably good screening at that point for the hepatitis C virus.
Right.
In blood transfusions.
I think it was still one of the non-A, non-B at that point.
Yeah.
Right.
Yep.
Right.
Sure.
So, based on this scorecard that I kind of gave you, the authors make a case for maybe the criteria by which the Hepatitis virus and the PEGI virus genera were defined taxonomically. Maybe that's going to have to be revisited as more sequences come. And in fact, Amit in his email said that there's a couple more papers that are coming out from the UK and samples from, I think also from the US, where a couple more human Hepatitis viruses are going to add to the diversity that we know about so far. So then there's a couple of things that we don't know in this. We don't have the transfused blood to find out whether the virus, the human Hepatitis virus, was in the transfusion itself, or whether these patients got the virus through some other way during the course of their treatment. And they don't have evidence of seroconversion in these individuals because at the point when they wrote the paper, they don't have a serological assay for the virus yet. But he states in the paper that they're working on that. They had these 40% unclassified sequence reads that I mentioned. They went back and looked through those to see if they could now find the human Hepatitis virus sequence in there, and indeed they did. So that suggests that there's probably, and I didn't really pay attention to the details on that, they probably used some very specific Hepatitis virus primers, and probably the samples were a little bit divergent from the human Hepatitis virus that they isolated, because there's some allusion to human Hepatitis virus diversity. Anyway, they talk about the fact that this doesn't seem to be as common a virus in the blood as the hepatitis C or the PEGI virus, which from that figure 1, at least in those 12 subjects that they looked at, both viruses were present in 7 out of the 12, whereas they only found these viruses in 2 out of 116 transfusion subjects and 2 out of 106 hemophilia patients. So they give a couple of reasons why that might be. Maybe the virus just isn't that prevalent, or maybe it doesn't establish persistence as frequently as Hepatitis and PEGI, or maybe the PCR screening method for viremia didn't amplify their more genetically diverse variants of the human Hepatitis virus. And
now,
they
of course, once you get a serological test for this, you could do a much wider study to look at some of those questions.
Right. And they point out that this inability to amplify a diverse population historically led to an underestimate of hepatitis C prevalence until its diversity was well known.
Right.
So, and as Alan said, you can do lots of things now with the sequence. You can study its prevalence, you can study its disease association and its persistence. But what I really wanted to know is whether they're working on synthesizing the full-length RNA to get a virus. And Amit said, yes, but we want to confirm the sequence first before we do that synthesis. That sounds like a good idea. So I thought it was a pretty cool little snippet. And I just wanted to give one more plug about Amit because he's in this new position. And so I asked him what he's working on now. And he has 2 focuses in his lab. He's working on developing animal models for rodent homologs of human viruses, including hepatitis C. And in fact, he's looking for postdocs in that area. So could give him the TWiV bump if you're interested in doing something in this virus discovery rodent model development. there's Amit's lab at Ohio State. And the other thing he's working on is some more new human and animal viruses found in samples provided in the U.S. and some international collaborations as well. So
this is an interesting group of viruses. Now, if you're wondering where GB comes from, GB virus C, this used to be called hepatitis G virus. It was discovered in 1966, isolated from a surgeon with the initials GB, who got sick with non-A, non-B hepatitis. So in 1966, we knew about Hep A and Hep B, but not any other hepatitis viruses. And this was thought to be the virus that caused his illness, so they called it initially hepatitis G virus and then GB virus C. And as Kathy said, there's also GB virus A and B. And they're all PEGI, and they're all PEGI viruses now. It's been classified as that, and they've since been isolated in other animals, as you heard at the top of this snippet.
This is from back in the day before surgeons wore gloves.
But it turns out that his hepatitis was not caused by this virus, and as you heard, it doesn't cause much of any human disease at all.
Right.
So what they're doing, in fact, when I was in Wisconsin way back in 2013. I did a TWiV with a couple of investigators there, and one of them has a program on these PEGI viruses and did virus isolation in Uganda from various monkeys, and that was a cool story. But my host back then just published a paper in Science, Adam Bailey, called Durable Sequence Stability and Bone Marrow Tropism in a Macaque Model of Human PEGI Virus Infection. So they can get GBVC to infect macaques, and they use that as a model to study the pathogenesis. And we may do that paper at some point. But GBV has also another nickname called the good boy virus because it apparently protects HIV-infected individuals from developing AIDS to a certain extent. This has been an association for which the pathogenesis is unknown. So this is sort of part of the reason why people have been interested in looking in wild animals for these viruses as well. So GBV, named after that surgeon, and it didn't even cause his illness, but he got it named after him. That's quite interesting.
Yeah, they took his serum and they injected it into tamarins.
Yeah.
And the name PEGI comes from, PE comes from persistent, and the G comes from GB, his initials, or hepatitis G, but anyway, that's where the PE and the G of the PEGI viruses come.
Yeah. Interesting story. Also, what I found interesting here also is this, there's these single-stranded DNA viruses. Everybody's got them, and you can see in this study that they're highly prevalent, and certainly over 95% of the human population is infected with these. And I remember walking around with Eric Delwart years ago in San Francisco, and he was bringing this up. He said, every blood sample we look at has these little single-stranded DNA viruses in them, and they don't appear to be bad for most people. And that's confirmed here. It's pretty cool.
Their biological function is to serve as positive controls.
That's right. That's right. Now, the other thing I think is cool here is that this whole thing came out of some reads that were really low similarity to known viruses, right? And that's what they picked up, as you said, and pulled out these new viruses. So all that dark matter, which you can't recognize, you just push it a little more and you can get something out of it. Yeah. That's pretty cool.
And the bottom line on all of this, though, is don't panic.
Right.
I mean, yes, they found a new virus that has been in human blood and is probably still in human blood at some frequency, and at some point in the near future, we're going to find out how prevalent it is. But it's not associated with any disease. There's not any reason to believe that it's harmful. It may turn out to be in some number of cases, but it's clearly not some huge public health threat. Well, as far as we can tell.
In fact, whenever you get blood, you're getting viruses. There's just no way around it. And Eric Darwell said if we threw out all the blood with viruses in it, we wouldn't have a blood supply. And they're largely harmless as far as we can tell, because we all have them and you get more of them, and most people it's okay. So yeah, don't panic. And that should probably be part of the title, I guess. Yeah. Either that or something to do with Peggy, because I kept hearing Peggy. Yes. Wasn't there a rock song about that?
Peggy Sue.
Peggy Sue. All right. Thank you, Kathy. That's a cool paper. I used to see Amit quite often back when he was at Columbia. So good luck out there. And I said to Kathy, he's where Stephanie Neal is, who was on TWiV a couple of weeks ago, but she's 96 miles away from him. So Ohio State is spread out.
Right. But you have a typical New Yorker's view of
that's
everything.
nothing.
Everything beyond the Hudson is just
I figured Ohio is Ohio, right?
Yeah.
Yes, you're right. All right, we have one more paper for you. This was sent in from Steve. Here's a fascinating article on origins of HAV that just appeared in PNAS that you might consider for a future episode of TWiV, Hepatitis A virus. And it was sent by Steve Backenheimer, who is a professor at the University of North Carolina, Chapel Hill, in the Department of Microbiology and Immunology, which we have had quite a bit of here on TWiV lately.
I have to say hi to Steve. I was on study section with him ages ago. When I was first an assistant professor, I was the postdoc fellowship study section.
Yeah, nice to hear that he's listening. Mm-hmm. Absolutely. So this is hepatitis A virus, which, of course, we have talked about because, as Kathy mentioned, it has a mere 121 targets sequence in the 5' non-coding region. It's a cellular microRNA, a liver-specific microRNA that's required for its replication. We have talked about that before. And also more recently, Lemon's finding, who's also at UNC, that the virus is sometimes enveloped in the blood. It is enveloped, and that may help shield it from antibodies. So that's another TWiV we did not too long ago. So this is a pretty cool virus. It's a picornavirus. In the same family as poliovirus, but a different genus, the hepatovirus genus. Oh, and by the way, the question here is, where does this virus come from? And in fact, the previous snippet that Kathy just did, that was the email too. Where did this virus come from? And
probably something you ate.
Who knows? But
further back, yes.
This paper is the way to answer the question, as we'll see. So you could do a similar study with Peggy virus, perhaps, and ask where it came from. So we don't know where that virus in our blood came from. But
so would you say its origins are shrouded in mystery?
Yeah. So hepatitis A was shrouded and is shrouded, but it's no longer mysterious. But certainly Peggy is shrouded in mystery.
Which is a phrase from this paper that...
Yeah, I liked it. That's why I put it up there.
A little dramatic.
Very dramatic. But people are getting very dramatic with their writing these days. I don't know. Anyway, where did hepatitis A virus come from? It is a virus that humans acquire. Of course, it's a plus-stranded RNA virus with a naked icosahedral capsid. It is transmitted by the fecal-oral route, and it replicates in your liver. It's shed in your feces, and you spread it to other people that way. It gets into your blood, and in your blood it has this cloak, this membrane, which it normally doesn't have when it's shed in your feces. It has tropism for your liver. You get infected. You have an acute period of jaundice and hepatitis, and then you typically recover, and you have immunity to it.
And it's typically, the fecal-oral route is like foodborne, waterborne type of infection, typically.
Right.
Something to think about as you mull over your Thanksgiving dinner.
Well, at this point, your leftovers.
Your leftovers, yes.
Yes, I had turkey sandwich for lunch, as I will have for the next 2 weeks probably. Until I asked my daughter if she wanted a turkey sandwich. She said, I have a turkey sandwich every day. Oh, I forgot. Yes, I should know that. Back to hepatitis A virus. So they want to know where it came from. This is a human pathogen. Where did we get it from? Because, as you know, most of the viruses we have probably came from some animal. So they did a wonderful study where they looked for HAV-related viruses in specimens, including tissues, blood, and feces, collected all over the world. 15,987 specimens from 209 species of mammals in 5 different mammalian orders. Now, listen to these orders. Are you ready for your marching orders? Rodentia, rodents. Scandentia, tree shrews. Chiroptera, you should know that. Bats, because we talk about bats a lot. Eulipotyphila, hedgehogs and shrews. And Afrosauricida, which are tenrecs.
Which look a lot like hedgehogs.
Yeah. In fact, they have these little icons for these animals in this paper.
Yeah.
And some of them are clearly rodents or bats.
Or people.
Or people,
Yes.
right. But I guess the shrews are kind of right. The hedgehogs I can recognize. They're kind of bulky, and you can imagine them being spiky. But some of the others are not easy. So they had all these samples collected, and they looked for them by doing first PCR with targeting conserved sequences in the virus genome, and then asking simply how many are positive for hepatitis A virus. So out of all these 15,000 plus samples, 117 were positive from 5 continents, 28 different non-primate hosts, including 13 bat species, 13 rodent species, 1 shrew, and 1 hedgehog species.
This is a boatload of work.
It's a lot of work and a lot of authors. Did I say who the authors were? No.
Probably don't want to list them all, but
Drexler is the first one. But in there is, and the last author is the Hepatovirus Ecology Consortium.
Which is probably a bazillion authors, yes.
And then before that is Christian Drosten, who we've talked about before on TWiV with involvement with MERS coronavirus. Stan Lemon is on this paper. And a number of other people that I recognize. Yeah, it's a lot of people, and it's from Germany, it's from Rotterdam, the Netherlands, it's from Russia. We got Germany, Chapel Hill, Germany, and so multi-continent, a lot of people involved here. So now they have 117 specimens. They're positive. They can get sequence from all of these and start to ask, how are these related? I have this incredible figure 1. I don't know if this is open access. Do you know, Alan?
I think it is not. You emailed a copy, but I had also logged on.
Yeah.
I think it pinged me for a log on for this.
But this first figure is this incredible assembly of all this information. They have one panel where they show a map of the world, and then all the places where the animal samples come from, and then for each continent, the Americas, Europe, Asia, and Africa, they have a pie chart colored as to what species of animal the viruses were isolated or the sequences came from. And so, for example, in the Americas, most of them were from bats, and then from rodentia, etc. So that's pretty cool. And then they have a phylogenetic tree where they arrange all of the sequences according to their relationship. It's a circular type of tree, and you can see there are 7 clades where they put all these viruses together, and they're colored again according to their species of origin, which is really nice. And at the lower right, there's red, which is primates.
This is one of those figures that, as a reminder, scientific articles are not like magazine articles. This is not some infographic that you glance at and say, oh, yeah, I get it. This is something that you're going to sit there and unpack for 30 minutes.
You could look at this a long time and get a lot. You could keep getting more information
out of it. Yeah,
because on this phylogenetic tree, for example, not only are they colored, but they have little icons. So if you don't remember that blue is bats, you can see a little bat. That makes a lot of sense, and you can. This is
like a compression algorithm to put about 50 pages of data into one column of one page.
And the green, that looks like a hedgehog, right?
Mm-hmm.
Yes.
You guys agree with that?
I think so.
And the orange are rodents, and so that's on this one. So this shows you there are 7 distinct clades. These viruses fall into 7 distinct regions, and they're somewhat geographically restricted. So, for example, you have the Mexican clades, and you have parts from other parts of the world as well that tend to cluster. So one of the interesting things about this is that the rodent viruses from North America, Asia, and Central Europe on this tree cluster very close to the primate hepatoviruses. And they all seem to have, they call this monophyletic, which means they have a common ancestor. 13 new hepatovirus species figured out from this. All right, on the next part of this that's kind of cool is that many of these viruses from specific locations are not monophyletic. They don't have a common origin. They seem to have multiple origins. Okay. In addition, different virus sequences acquired from different bat hosts globally have different phylogenetic positions, whereas from the hedgehogs and shrews, you have 2 phylogenetic positions on this map. Basically, what this means is that over time, these viruses, the non-primate hepatoviruses, have gone from host to host. They have done host jumps and spillovers in different species. And that gives you this complicated phylogeny, right? So it's not simply a virus entered rodents and has been passing through the rodents only for many, many years, but it may have entered a rodent, then it went to a bat, and then it went back to the rodent and to a tree shrew, et cetera, et cetera. It's rather complicated, which is pretty cool. Also, if you look at they have a lovely graph of the phylogeny according to host and according to diet. And on this phylogeny, if all the primate sequences can be seen to have come from a rodent of some kind. Okay, the primate ancestor is consistently a rodent host. And now all the mammalian hepatoviruses go back to a Laurocyatherian host and an insectivorous diet. Now, you guys knew what a Laurocyatherian is? Because I certainly didn't.
No.
There used to be a northern supercontinent before the continent separated called Laurasia.
Laurasia, yes.
And the animals that live there are Laurasiatherian animals, including shrews, hedgehogs, and others, pangolins, bats, whales, hoofed animals, and carnivorans, among others. So, these guys evolved when the landmass was together, and then as it separated, they went their own ways, but they're still Laurasiatherians. And anyway, that is the origin of a lot of these HAV sequences in mammals. But the thing is, whoever it was ate insects. So who knows? It could have come from an insect to begin with, right? We don't know that. Bats are the most relevant donors for many of these host switches, followed by rodents. And Eulipotifla, what is that again? Eulipotifla, hedgehogs and shrews. Anyway, lots of cool information from this. And finally, the primate donor did not involve a host switch. So once it got into primates, it stayed there. It hasn't gone to anyone else. So the basic premise that we learned from this is that there are lots of Hep A-related viruses globally in many different species. We didn't know this before. We just had basically human viruses. And this gives us an idea that these viruses have jumped around and that our viruses came ultimately from some kind of rodent host many, many years ago. So they entered humans a long time ago from a rodent host. Now, recently on a TWiV, someone said, or maybe it was at some other venue, I can't remember. It was when I was in Portland. They said, what's the origin of poliovirus? I said, I don't know, but this is exactly the kind of study that you would need to figure it out. You could do this with these same samples. You could look for poliovirus sequences and then say, was it a rodent virus at some point, or a bat or a shrew or whatever? It's pretty cool.
Hedgehog. I'm pulling for hedgehogs.
Hedgehog.
You're pulling for polio coming from hedgehogs?
I don't know. Hedgehogs are cute.
They are cute.
Yeah.
I think they're very neat.
But yeah, now that we have the sequencing tool, the technologies to do this are the sequencing tools and the bioinformatics tools.
Right.
You have to sequence and look for the viruses, and you have to be able to assemble the trees and figure out the relationships between them in order to narrow down where they likely came from. And the utility of this is if you do enough of these, you may start to see patterns that would help you predict where other viruses might come from.
Right. So this one figure is lovely. In fact, the last part is this nice, just a simple XY bar graph of the number of interorder host switches according to species, so bats or rodents or shrews, hedgehogs, and people. And you can see the most interorder species have occurred among bats, and finally, and then next rodents, and then the shrews and the hedgehogs, And then finally, people have had no inter-host switches at all, the primates. It's really nicely projected. However, that's not the whole paper. There's more. It's cool stuff coming. Because this, in many papers, this is it, right? This would be the sequence in the phylogenetic analysis, but they then go on to do some other cool things. They look at the genome organization of these viruses. They get 14 near-complete genome sequences from 9 different species. And Hep A is a typical picorna, has a long open reading frame with 5' and 3' non-coding regions, and you can see them here. You can see the structures of the 5' end. For example, the internal ribosome entry site is a type 3, internal ribosome entry site. And you can look at various parts of the open reading frame. You can look at protein structure. A lot of these are conserved from hepatitis A virus among all of these isolates as well. So the one cool experiment that they did, they wanted to know if sera from any of these animals would recognize human hepatitis A virus. So they had sera from 111 bats, 10 different species, 114 rodents, shrews, hedgehogs, and they simply asked, do these sera react with HAV particles? And only bat sera react in immunofluorescence assays with HAV-infected cells. And none of the others do, and this was confirmed by immunoprecipitation. They don't know if, in fact, this represents cross-reactivity, or they suggest a possibility is exposure of bats to human virus, contaminated environmental foods, and that sort of thing. But that's quite interesting. They're surprised that there are no cross-reactive sera from rodents, because there are quite similar rodent hepatoviruses and human HAV, but they may react differently to infection. Then they ask, in these animals, where is the viral RNA? So they looked at infection patterns in bats, rodents, hedgehogs, and shrews, and they find, by looking for viral RNA abundance in different organs and in situ hybridization for viral RNA, that the patterns are very similar in all of these animals. They are hepatotropic in all of these animals. They infect the liver, but they can also find them in other places, like in the spleen as well. So that's pretty neat that you can do this, not just have the sequence, but you can say this is a hepatotropic virus, just like human HIV. Then they did a neat experiment where they had a German animal shelter that had hedgehogs in it, and they took 24 of them and monitored them over a 220-day period. So 9 of these initially tested positive for hepatoviruses in 3 separate specimens, and these viruses were identical. The multiple isolates were all identical with them. Fecal shedding was observed over 79 to 142 days in 4 animals, and that's sort of in line with the timeline of fecal shedding observed in humans who are infected with HIV. And then in consecutive...
HIV.
What did I say, HIV?
Sounded like HIV.
Yeah, I think I slipped. HAV, getting really close with these letters now. In consecutive specimens taken less than 15 days apart. So remember, again, they have these 24 animals they can sample regularly, which is really cool. The second specimen always showed a significant decrease in virus concentration, and they're saying this is like an acute infection, which is Hep A in people. You get infected, you have a relatively quick infection period, and then you resolve it. So it looks like these animals got infected, and then they resolved the infection. In fact, 4 animals cleared the infection during the study period. So they were lucky that they caught them at some point in the infection, and 9 of them, and then they could see a couple of them clearing the infection as well.
And this also teaches us that there are hedgehog shelters in Germany that keep these animals while they're sick and then release them.
Yeah. Mm-hmm. And in fact,
which I had no idea.
They released these animals, and then they said these 9 infected animals were fine when they were released, so that it had low pathogenicity, at least, in of those animals. And then the other study they did, which comes under this, ecology pathogenesis, they had specimens obtained over 3 years in a German bat maternity roost. Okay, the females annually form a colony that lasts about 3 months. They give birth in a synchronized way. So they get together, make this colony. It lasts 3 months. They give birth, and then the colony disperses. So they were able to sample this colony. They got bat hepatoviruses and fecal droppings as soon as the colony formed in May 2008. Then a month later, the detection rate of virus in feces had already decreased. And then after birth in mid-July, the sampling rate went up again. And then before the bats left the colony, at the end of the month, the detection rate fell again. So they say there's no persistent infections probably, but waves of acute infections. Bats get infected, they have an acute phase of disease, and then it's cleared, and then others are infected as well. And they make the point saying that these waves of acute infections in this bat colony is reminiscent of HIV outbreaks in children's camps before we had an HIV vaccines. Kids would go to camps in the summer, and they'd all get HIV infection, waves of HIV infections. So that's pretty cool that they had these two opportunities to sample animals out there for these viruses to see what was happening with them, and they in fact found them without, these are not cases where they had to do any infections. They're just naturally infected, and apparently they're fine. So this shows you that there are multiple HIV-like viruses out there in many species over many continents. We think the origin of human HIV was probably a rodent host many years ago. They say that whether this introduction at the people took place in the primate stem lineage before the split of hominids and cercopiths about 25 million years ago is not known, because we don't have enough human strains, actually, from non-human primates. Sorry, we don't have enough strains from non-human primates. Otherwise, we might be able to tell when the introduction came into those individuals. And then we have some indication of this virus is a hepatotropic virus that causes acute infections in multiple species. So I think this is pretty cool that they used multiple approaches to identify the origins of these viruses. And as I said, they ought to do it for polio next.
Sure.
And Kathy's betting on hedgehogs.
Well, it's just that Lindsay Rollins had one or more hedgehogs when I visited her. She's Dave and Bryony's daughter many years ago. So I've seen hedgehogs.
They're quite small, aren't they?
Yeah.
They're very cute.
I have to ask Stanley. He's going to come by. Stan Lemon was going to come by in January if they're doing this with polio, because I think that would be really cool. Mm-hmm. And then my life would be complete, at least my virology career, and knowing where polio came from. Anyway, very nice paper. Thank you, Steve. I hope you liked it. And now we know the origin of Hep A.
Yes.
I just wanted to comment on one word in the discussion. Our findings render this picornavirus genus exceptionally speciose.
Speciose. That's great.
Yes.
There are lots of words in this that were needed lookups. For example, patristic distances.
Yeah.
So I look up the definition, and it says patristic distances, the number of apomorphic step changes. Oh, now I have to look up apomorphic. Oh, man. And what was the species word again?
Speciose.
Is that a real word?
I think so. Yeah. Like
Alan,
verbose.
You got to make a title with speciose. Let's see what you can do. Cool.
Yeah, we have speciose in the human Hepatitis too.
Yeah. Speciose and verbose viruses. All right, this episode is also brought to you by the ASM Biodefense and Emerging Diseases Research Meeting. You have until January 14th for early bird registration. And, of course, if you register by then, you save money. This meeting is going to happen in Arlington, Virginia from February 8th through the 10th. It's the premier event to discuss how to defend against the growing threat of bioterrorism and to shape the future biodefense research agenda. This meeting is not just about biodefense, though. They also talk about emerging pathogens. And I've been to this for the last couple of years. It's a pretty nice meeting. The keynote speaker is going to be Tom Frieden. And we will be doing a TWiV there. I cannot yet tell you what the topic will be. We've done them in the past couple of years as well. They're always interesting. So you should check that out. Go to asm.org/biodefense2016 if you're interested in registering. Early bird ends January 14th. Let's do
not to undermine an advertiser, and this is a very cool conference, but it's a growing threat of bioterrorism.
Yeah, I when I read that, I felt like making a snarky comment, but you cannot bite the hand that feeds.
No. As I say, it's a good conference to go to, but I would.
That may have been the first year. The first year of the conference, it may have been.
There's certainly a growing industry of biodefense. I would.
Yes, for sure.
With that.
Well, it was originally, I think, a biodefense meeting, and then they expanded it because there wasn't enough biodefense-related things to run a meeting. And what there was, some people just didn't want to talk about it.
Right.
Which doesn't bode well for a meeting. Nevertheless, we thank ASM for their support of TWiV.
And emerging diseases, yes.
Yes.
Those two things do overlap, and it is certainly a worthwhile subject area.
Okay. Let's see. Anthony sends us a virus weather report. That's his title.
Yes.
Sends us an article, epidemiologic and other analyses of human high pathogenicity avian influenza virus infected poultry flocks. This is a report from June of 2015, and he writes that this document discusses at some length wind as a possible factor in virus dispersal, hence the virus weather report.
Yes.
And this is well known for a number of viruses. I know foot and mouth disease virus is well known to be transmitted by the wind. Let me take Asal's email, and then we can move on. Asal writes, Dear TWiV team, I am a molecular microbiology lab technologist and an avid TWiV listener. When in 2010 I first discovered TWiV, I was so excited for being able to learn more virology by listening and not just by reading. Ever since, I have enjoyed learning both updated and basic virology through your highly informative podcast. I started listening to TWiV while staining shell vial cultures in our BSL-3 lab at MedFusion in Texas, and now I've been mostly listening while driving in slow traffic. Yeah, I know all about that. I would recommend listening to TWiV to everyone who is not only interested in viruses, but also interested to see how scientists share and exchange their opinions, ideas, and knowledge with respect and humor. Is that us?
I think so.
On TWiV episode Cat in the Heart, yes, the cat in the heart, there were questions raised about screening male patients for Pap testing and HPV. It's been almost 2 years that we at our molecular ID lab performed FDA-approved HPV DNA testing by Hamilton Roche Cobas 4800 system on anal and pharyngeal Pap vial collections from the male population. The reverse HPV Pap algorithm has also been recently approved by using the same platform, and provides a link to that. The assay tests for HPV 16, 18, and other high-risk HPVs, including 11 subtypes. We sometimes attain triple positives on anal rectal collection. Sample may be preserved and transported in vials. I also had a question regarding the OPV and IPV used for eradication. I am wondering if any country has ever used them in combination in a way that IPV has been followed by OPV to reduce the risk of vaccine-related paralysis associated with OPV. And the answer is yes.
Yes.
In the U.S., in fact, in 2000, before we switched to IPV exclusively, there was a transition period of IPV followed by OPV. Right, Alan?
Yep.
And that's been done in other countries as well. But, as I will tell you, you don't want to have both always because that makes it more expensive and difficult. So at some point you have to just switch to IPV. When I was a junior in high school in Iran, the eradication program took place in my hometown, Tehran. In my vague memory, I recall my parents took my youngest sister twice to the vaccination stations within a year or two, and I remember my father saying they gave me the oral one this time, referring to the second round. At the time, I did not know enough of the fascinating world of viruses, so I didn't pay attention to the details or how it was carried out then. My parents do not recall it either. In 2001, during my second year of an undergraduate studies in microbiology, I took my virology course taught by Dr. Talat Mokhtari Azad, who had conducted the polio eradication plan in Iran. After reading Vincent's interesting and concise article about OPV, IPV, and the history behind them, it became a question for me whether the eradication in my hometown was really ever conducted in a combination format. I have tried to get in contact with my former virology professor, but have not received a reply from her yet. Do you know of any eradication plan in any region that was administered using both OPV and IPV? At the end, thank you for your wonderful show. Virologists are admirable scientists, and the ones who spread the knowledge on viruses and microbes to the world will be remembered in the history of social media and virology.
Wow.
Keep up with the great show. P.S. Today, October 6th, is partly cloudy, 18 Celsius here in Colleyville, Texas. Kind regards, Asal. So here, I don't know that any eradication has done both. I just know so far it's been mainly OPV, and now, of course, in this transition phase, we're starting to combine the two, but only recently. And here are the current WHO recommendations for global immunization. This is in their February 28th, 2014 weekly epidemiological record. First, all children worldwide should be vaccinated against polio. Second, for countries that use OPV only, at least one dose of IPV should be added to the schedule, so you get 3 OPV plus 1 IPV. Where vaccine-associated polio is concerned, you do a combined IPV-OPV schedule. And then there are countries that can use IPV only, where you have high coverage and low importation risks, such as the U.S. and Canada, etc. So there are cases where we are using combined immunization, but up to this point, I would not say that it has resulted in eradication anywhere. It's been mainly OPV. Would you agree with that, Alan?
Oh, yes. I mean, when you say an eradication campaign, there's kind of a fuzzy line between an eradication campaign and a vaccination campaign.
Should be vaccination, yeah.
Right. We have an ongoing vaccination process in the developed world. In the U.S., for example, IPV is now part of, it's actually incorporated in one of the combination vaccines that kids get. So it's just part of the schedule. And is that an eradication program? Well, not exactly, but it's kind of a keep it eradicated program.
Well, I think as you know, there's some countries that have or some regions where it's been declared eradicated. Right?
Yes.
So I think that the writer is asking, in those areas, has the combination of the two vaccines resulted in that
responsible state? Has the combination been used during that transition period from polio present to polio absent? I don't know of any.
Yeah, I don't either.
I think it's generally that happens in an area where you're doing OPV only campaigns.
Yeah.
Just because of the logistics of it, and OPV only is the most common protocol in the countries that still have polio.
Yeah. Okay, Kathy, can you take Eleanor's?
Eleanor writes, "Hello all. I hope this can be considered a follow-up, even though I'm a week late. In your discussion on mucosal immunology, you wondered why inactivated flu vaccines, given systemically, are protective, since flu infections occur at a mucosal site. I think it's in large part due to this. Normally you think of IgA as the mucosal antibody, but in the lungs there's actually a lot of IgG, which is transduced from the blood across the epithelium onto the mucosal surface. This happens in normal lungs, but even more so if there's inflammation. In the upper respiratory tract, on the other hand, there's plenty of IgA and not as much IgG. I don't know what the situation is in the gut, but perhaps IgG is not transsudated in the same way there. I also wanted to say that I was glad to hear you guys discuss feline immunodeficiency virus recently, as well as about Stephanie's coronavirus research in pigs. I'm a VMD PhD student, and it's great to get more exposure for work on viruses that are relevant for both animal and human health."
Cool.
Mm-hmm.
So that's how you say it, VMD, not DMV. No, what is it?
Well, it would be
DVM.
It would be DVM.
Yeah.
But
this way it's like
VMD. So
VMD.
maybe she's not in the United States.
I don't know.
You know.
Yeah, but this would be veterinary MD, right? Which would make sense also. Interesting. All right, let's do one more. Alan.
Yes, John writes, "Dear Twiverati, I hate writing in critically because you all are so wonderful, but I had a couple of critiques, questions, problems regarding some points of discussion in TWiV 361. The problem is, when everything's fine, writing in would waste your time. Anyhow, there were a few nagging things about your Ebola virus transmission discussion that I couldn't get past. If someone else has written in about any of these, feel free to skip that point. Here they are. First, Now, this was the paper, the report of possible first sexual transmission of Ebola virus. So first, directionality of transmission from patient S to P or P to S. As far as I understood the paper you were discussing, P first experienced Ebola disease symptoms 7 days after her sexual encounter with S. You said it was possible that P transmitted to S because the incubation window would accommodate those 7 days. I recall Tom Frieden and many CDC representatives repeatedly saying that it's impossible to transmit EBV during the incubation period. Has that been overturned? If it hasn't, it is hard to see how transmission could have flowed any other way than S to P, or, and I think this is more likely, some other person who's totally unrelated gave it to P, and S's one positive is just the contamination that Alan suggested. John, you're absolutely correct, and I believe I am the one who screwed that up, so my apologies.
But you had some co-hosts who didn't.
Nobody else called me on it.
No.
But yes, as I was looking at the data in the paper, I said, wait a second, this timeline doesn't look right. And I still feel the timeline doesn't look right, but the directionality, from the evidence we have, the directionality could not have been from her to him because she was asymptomatic, and that is correct. If you're asymptomatic, you're afebrile, you are not capable of transmitting Ebola virus. That is still correct, and I screwed it up.
Yep.
So, but yes, could have come from somewhere else, and I stand by my confusion about the who got what from who. Second, if sexual transmission of Ebola virus were likely, it would be the next HIV. I think was something said. No, if HIV had an infection cycle similar to EBV, it wouldn't even be the current HIV. The reason HIV is so efficient and dangerous is that it persists in patients as an asymptomatic infection for a period of years, which increases the chances of sexual transmission to multiple people. It was also a totally unknown agent to medical science for decades of its initial spread, during which it got into the transfusion blood supply as well, where it spreads much more readily than by sexual activity. In order to be, quote-unquote, the next HIV, Ebola virus would have to persist in semen for years, or it would have to be so common for it to be transmitted sexually that this route would have been obvious from the epidemiological data, right? And even then, it kills so readily and quickly that I doubt it would achieve HIV levels of prevalence. I suppose a third option would be for there to be a high number of asymptomatic carriers of Ebola virus, but I don't think there's any evidence for that either. I partly agree. I think what we were getting at with was that the argument is increasingly being made that Ebola virus persists in semen, possibly in vaginal fluid. And there's this, in the paper about sexual transmission, this notion of it transmitting sexually, which I still don't think has been quite proven. But if it were the case that this persists in those fluids as a viable virus for months and months and months, which is what the PCR data are being interpreted to mean, then I think the point of it would be the next HIV is this would be a much more prevalent virus. This would be much easier to catch than it is. I think that's kind of what we're getting at.
Mm-hmm.
But yes, good points about HIV is in a very different situation because of the long incubation asymptomatic period. And also with HIV, during the asymptomatic period, you can transmit it. All right, John's third point, Lassa fever. Patient S repeatedly tested negative for Ebola virus while he was ill with an unspecified hemorrhagic fever, which he survived. Also common in West Africa is Lassa fever, which infects hundreds of thousands of people annually, and in 20% of them causes a hemorrhagic fever. The mortality is much lower than for Ebola virus. Was patient S tested for Lassa virus during his illness? I think that is an outstanding question.
Mm-hmm.
That is a very real possibility.
We don't know the answer, no.
Right. Fourth, Rich's comment about SSPE and paramyxoviruses. This is rather close to my PhD work, so of course I took a lot of interest in that comment. Interestingly, persistence of infection has also been observed in Nipah virus infections, with reappearance of symptoms months after their apparent resolution. Persistence of paramyxoviruses is thought to rely on a few things: the ability of the surface glycoproteins to fuse adjacent cells when expressed on the cell membrane, forming syncytia, and mutations in the matrix protein that prevent typical virion budding. It's my understanding that Ebola virus has to enter an endosome in order to fuse, and it relies on the cholesterol transporter NPC1 to facilitate fusion within endosomes. It seems fairly unlikely to me, then, that cell-to-cell fusion can be facilitated by Ebola virus, and that seems to make a mechanism like the one seen in SSPE less likely, doesn't it? That's not to say there isn't another mechanism of Ebola virus persistence or that Ebola virus doesn't find some way to cause cell-cell fusion. I just think the hypothesis of a parallel to paramyxoviruses is less likely than other explanations. Of course, I might just be wrong about Ebola virus dependence on endosomes for fusion. Anyway, I hope these aren't too nitpicky or combative. It was a great episode, which is why I ended up with so much to say. To end on a positive note, Vincent's Rocky Horror Picture Show reference, see Androids Fighting, made me laugh out loud.
Well, I'm glad someone got it.
And that's from Leland Feynman, a.k.a. John Schuyler.
Who was on TWiV.
Yes, who was on TWiV. P.S. Oh yeah, I defended my PhD since my appearance on TWiV, so there's that. What's next? Not sure, but excited to find out.
So back to point 3, Lassa fever.
Yes.
So if patient S in fact had Lassa fever, then there's no sexual transmission to P, right? And P got it some other way.
Got it from some other source.
Yeah. Because P was certainly diagnosed with lab positive Ebola, right?
Yes, I have no doubt that P had Ebola virus.
Yeah.
But the data are pretty clear on that. I have some question about whether S had Ebola virus before contact with P.
Yeah. I just wonder if in these Ebola treatment centers, if they just look for Ebola virus or if they look for Lassa as well. You know?
I don't know.
I don't. I suspect that they would not, but I don't know the answer to that. So those are all good points, John.
Yes.
No, not too nitpicky, not combative. It's fine if we are wrong.
Totally appropriate.
It's okay.
Yes, particularly your first point was a very important correction. I'm glad you brought that up.
All right. Let's do some picks of the week. Alan, what do you have?
I have something that came up a little while ago. This is on the blog of Bill Gates, which is not one of my regular reads, but Bill Gates, after infecting the world with computer viruses, after building a company that makes some kind of operating system, of course, has now been fighting disease, and the Gates Foundation is huge in public health and the polio eradication effort. Bill is apparently also a fan of Randall Munroe, who does the webcomic XKCD, a few of which I've linked as various picks of the week. And this is, so on his blog, Bill Gates posts this XKCD comic, which is about the polio eradication campaign, and it's quite fun.
Yeah, it's good. I've seen this. It is very good. How often does Bill blog? Gates
don't
notes?
Know.
Do you think he writes them himself?
I was wondering that. I don't know.
It would be nice if he did, right?
Sure.
He didn't have to, but...
he can do whatever he wants.
Yeah. Kathy, what do you have?
I have something that I need to thank Adam Loring and Sarah French for. When Matt Freeman was here, Mike Imperiali, Adam, and I went to dinner with Matt, and somehow tardigrades came up in the conversation. And so just last week, the tardigrade genome sequence came out in completely sequenced, and it's in PNAS. It's one tardigrade genome sequence. And then I was telling Sarah French about it, and she found this cool link to a video that's to a science alert site that has a video that's constantly running. There's no sound, and you can see the tardigrade swimming. And and that thing in the center of its face comes out periodically while it's swimming, and it's just amazing. Anyway, so the interesting thing about this tardigrade sequence is how much horizontal gene transfer there is. And the tardigrades are these things that are highly resistant to radiation and desiccation. They can live for 10 years. And so the authors speculate that when they dry down, there's still 3% water. And then when they hydrate back up again, well, their DNA is all fragmented, and when they hydrate back up again, there's some holes in pores or membranes or something, and that's when DNA can go in, and that's what leads to this really high rate of horizontal gene transfer. So on average, most organisms have around 1% of their DNA that can be accounted for as coming from other organisms. And then there's a rotifer that was previously known to have maybe around 8%, and it was the record holder until this tardigrade, which is around 17.5% foreign sequences from bacteria. I forget what all other things. It's in this Science Alert thing, which I clicked away from. Anyway, so it was just really cool. And then we had a little email exchange about it, and we had been talking about planetary protection, and these things can survive the harsh environment of space. And so Matt was proposing that you could use these things to try and figure out if there's life on Mars, because you could expose them to Mars and then sequence them before and after. And if they picked up sequences, then they would have to be from Mars. But as Mike pointed out, that you would be assuming that the life on Mars has DNA and so forth. But anyway, it was pretty fun.
And also you'd be contaminating Mars.
Right. And so this came up because Mike Imperiali is now on the NASA Planetary Protection Subcommittee. So we had talked about that at dinner as well. And some of the probes that have gone to Mars were not as thoroughly sterilized as some of the earlier probes that went to space.
Oh, so it's too late.
Maybe so. And one of them is particularly close to one of those streams of water that was recently identified.
Yeah, we already messed it up.
Yeah.
Not surprised, are you?
No.
They get DNA from bacteria, plants, fungi, and archaea.
That's right.
Amazing.
Yeah.
Yeah, it's pretty cool.
Are you still watching the guy swim? I am.
He's fascinating, yes.
This is really cool.
Yeah. And then in the show notes, I put the picture of the actual one that was sequenced. He's kind of happy looking, and you can actually see his eyes in one of the pictures, but in the scanning EM, the eyes kind of disappear.
But he's not happy after the scanning EM.
No. And the common name for these things are water bears. And they're about can be about a millimeter long. So.
Which is cool. Good, because if they were a meter long, we'd all be running from them.
Mm-hmm.
But Dixon has pointed them out in some pictures and things that we've had. So we've talked about tardigrades before.
Sure.
They get into everything.
All right. Here, today I'm at home, so to get my pick, I just turned around to my bookshelf, and I picked a book I read some time ago, and I've always been meaning to mention it, but forgot about it. Out of sight, out of mind, I suppose. It's a book by Charles Kalischer. It's called Lifting the Impenetrable Veil, From Yellow Fever to Ebola, Hemorrhagic Fever, and Sars. So Kalischer has had a very long career in virology. He was at the CDC in Fort Collins for 27 years, chief of the Arbovirus Reference Branch, director of the WHO Collaborative Center for Arboviruses. He's now professor emeritus at Colorado State University in Fort Collins and a member of the Columbia University Center for Infection and Immunity. He's a pal of Ian Lipkin. So he wrote this kind of personal account of the history of one part of virology anyway. It's a rather thick paperback book, and it's full of a lot of pictures of him with various people over the years and other virologists of various sorts. And I'm looking here, I see Charlie Rice is here, and him with all sorts of people. So this is his view of virology, which is interesting because everyone has a view, and someone in the field so long, it's kind of neat to hear what they think about things. So cool. It has some very good parts and less good parts, but the very good ones are interesting. So check it out. Lifting the Impenetrable Veil. A little bit dramatic, but, you get to write your book. We have 2 listener picks. One is from Judy, our friend in San Diego. She sends a link to Jennifer Doudna's TED Talk, and this is entitled, "We Can Now Edit Our DNA, but Let's Do It Wisely." So a good companion to our discussion earlier. And the bio says, Jennifer Doudna was part of inventing a potentially world-changing genetic technology, CRISPR-Cas9. So that's fair. That's fair. I met Jennifer Doudna many years ago. Listen, this is funny. I was at a Gordon conference, and she was a graduate student.
Mm-hmm.
And she was there, just a normal person. And now she's going to get the Nobel Prize, personally. What were you going to say, Kathy?
We had her here for a seminar series where we had outstanding women in science shortly after I moved here. And, it was way before this, so it was pretty interesting.
I met her and Emmanuel Charpentier at the Janssen Award symposium at the New York Academy of Sciences last year, which they were getting that award, obviously. And I actually I talked to both of them about TWiV and to see if they'd be interested in coming on the show, but that was before they became even bigger hits. So they certainly don't need the TWiV pump.
No, but it's always interesting to have people on and talk about
I think
stuff.
I think they'd be good guests if we could, get penciled into their schedules.
I watched the TED Talk. It's only about 15 minutes long, and it's good.
Oh.
So, people who want to know a little more about it, it's, I think, very accessible.
We have one more listener pick from Ramon, who sends a link to what looks like a lot of stars. Anybody know what you do with this? I clicked around for a long time, and it looks like
you can scroll it.
You can zoom in. There are settings. But I'm not sure. You can move from left to right, and there's part that's purple. Maybe that's where we are. Maybe that's maybe this is the Milky Way. I'm not sure. But it's ggs.astro.rub.de, and there's
quite a. Is this a view of the entire sky?
Maybe. Oh, here we go. No, that's just the. Oh, I see. This is.
Once you zoom in a little bit, it gets. There's a lot there. There's a lot of depth to this, and you can scroll around for quite some distance.
Yeah, maybe it's the entire. What did you say it was?
Maybe a view of the entire sky?
Sky, maybe, yeah. It's served on one of these high-resolution image sources, so you can keep zooming in over and over again. I don't... Yeah,
there is a limit to the zoom. You can't, see the little green men's eyes, but...
and
but
there's settings. You can set contrast and gamma and all this
stuff. Might be something to play with for
play with it, and you can make it full screen, and I don't know. It's kind of cool, but... it would be nice if there were some documentation.
You can choose from some options I don't understand.
But Ramon just sent it to us. Okay, thank you, Ramon.
Okay, thank you, Ramon.
And at the end here, I want to announce another contest to give away some 2-volume sets of Principles of Virology, the 4th edition, which is really nice. It's paperback 2 volumes. It's all updated. Here's what you need to do. You don't have to go onto any social media. You do have to use email to twiv@twiv.tv, and you get 280 characters. Twice as many as Twitter, and 280 characters or less. That includes spaces. You write about why you listen to TWiV.
Twice as many as Twitter, so it's Twitter.
Yeah. twiv@twiv.tv, and we will judge the most creative entry. And the next time we're all together, maybe we'll do the Twitter ones that we got. I'll list them all here, and we can figure out which one deserves a book set. So that's the second. And there'll be a couple more. All right. This one, you just have to listen to the end to do. There's no social media. Right. Mm-hmm. You can find TWiV at twiv.tv, also on iTunes. And if you want to send us questions or comments, twiv@twiv.tv. And try your hand. It's only 280 characters. That goes by really quickly.
Yes.
But it needs to be creative, interesting. The ones I've gotten so far have been good. All right. TWiv@twiv.tv. Did I say that?
Mm-hmm.
I think so.
Doesn't hurt to repeat it. TWiv@twiv.tv. Kathy Spindler is at the University of Michigan, which is in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This is a lot of fun.
And Alan Dove is at turbidplaque.com. And he's also on Twitter as Alan Dove. Thank you, Alan.
It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.de. TWS. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.